Hi, welcome for today's session. Here we will be looking at cell organelles. We will continue looking at cell organelles. As we saw, we have a number of them, and today we will devote our time in, first of all, reminding ourselves that much of the cell is devoted to making proteins. Much of the cell is devoted to making proteins. So in this case, if we look at proteins which are made of 20 different amino acids, they will have unique characteristics. Then for them to have unique characteristics, many of the proteins must be made, and these proteins should be unique in different sizes and polarity and even acidity. They can form very long or short chains depending on the need of the protein. So, and multiple protein chains can also interact with each other. This almost limits variety of shapes and interactions, making proteins very powerful. So proteins carry out many critical functions, so they need to be made correctly. Proteins need to be made correctly because many of the enzymes that we know carrying out functions or uh, aiding in chemical reactions. So for example, enzymes are proteins, and we saw that if they lose their structure, they cannot perform their function, and this tells you the importance of why the cell should always be able to make proteins which are unique and that carry out the specific function without making any errors or mistakes. So here we are having nucleus. The nucleus is found in the center of the cell, as we saw earlier, and is the storehouse for most of genetic information. It is the store for genetic information and also controls cells activities. It has in it the DNA, deoxyribonucleic acid. The DNA, of course, is responsible for holding the genes. So DNA contains contains genes that are instructions for making proteins. That are instructions for making protein. So what do you think will happen if, for example, there is an alteration or a change in the structure of the DNA? Directly, any change on the DNA structure will directly alter the genes, and therefore all the proteins that will be made will be different from the required proteins. So there are two major demands on the nucleus. One, DNA must be carefully protected. One, DNA must be carefully protected. That is a very essential demand because we have seen that the DNA is the one that contains the genes that are instructions for making proteins. So any alteration on the DNA will basically alter all the proteins, and this will be, of course, disastrous to the organism. Two, DNA must be available for use at the proper time. Must be, should be DNA must be available. DNA must be available. DNA must be available at the proper time. This means that when it is required, it should be available. So molecules that would damage DNA need available for use at the proper time. So molecules that would damage the DNA need to be kept out of the nucleus, and for them to be kept out of the nucleus has unique features. I mean, the nucleus is surrounded by a nuclear envelope which is made of two, two strands. So we say molecules of the DNA need to keep out the nucleus. So, but many proteins are involved in turning genes on and off. So we have these genes that are meant to turn proteins on and off, and therefore allow access to the DNA. So special structures of the nucleus help it meet these demands. So we have these proteins that control what really is allowed in or out of the cell of the nuclear pores. So we have nuclear envelope, which protects the inside of the nucleus, and we have nuclear pores that allow only specific materials to pass. So even the required genes are only copied when they are required, and when they are not required, they are not copied. So the nucleus is composed of the cell's DNA enclosed in double membrane called the nuclear envelope. Each membrane in the nuclear envelope is similar to membrane surrounding the entire cell. So if we are to look at the picture of this is what it looks like. So we are having here the nuclear envelope surrounding the whole of the nucleus. When you cut through to see through, you, you can see this is the nucleolus, and this is the inside of the nucleus. And the membrane has these nuclear pores. So here we have nuclear pores. Here we are having the nucleolus. So as you can see them there very clearly, These organelles we also have organelles which are made inside the nucleus. So inside the nucleus, we have the nucleolus. The nucleolus functions by producing, so the nucleolus is a dense region where tiny organelles essential for making proteins are assembled. Where tiny organelles essential for making proteins are assembled. For making proteins are assembled. And these are ribosomes. So the ribosomes are assembled in the nucleolus, and they are just but a combination of proteins and RNA molecules. Ribosomes are a combination. Ribosomes are a combination. Ribosomes are a combination of protein and DNA. So what makes up the ribosome is nothing but the DNA, proteins and RNA, but not rather DNA, sorry, and RNA. So that is what makes up the ribosomes. So we will be discussing them in the next unit, where we will be looking at their functions. So we will look at them in details. The next thing that we are going to look at here is the endoplasmic reticulum. So endoplasmic reticulum of most eukaryotic cell is filled by, almost the whole part of the cell is filled by endoplasmic reticulum. So the entire part of the cell is filled with endoplasmic reticulum and ribosomes. So when you are looking at endoplasmic reticulum, you have also to look at ribosomes, because normally ribosomes, some of them are attached to the endoplasmic reticulum. So endoplasmic reticulum, or ER, is an interconnected network of thin folded membrane. The composition is similar to that of the cell membrane and nuclear membrane. The endoplasmic membranes form a maze of enclosed spaces. They form, so it has, ER is an interconnected network of thin folded membranes. Is an interconnected network of thin folded membranes. It is an interconnected or network of thin membranes. The composition is similar to that of cell membrane and nuclear membrane. So numerous processes, including protein synthesis and lipids, occur both on the surface, numerous processes, including protein synthesis, including protein production of proteins and lipids take place on the surface and the lumen, and inside the lumen, inside the lumen of ER. So inside its lumen, these processes can take place. ER membranes have many creases and folds. So if you have ever gone through camps, you will discover that the sleeping bag you are carrying might look very small, but when you open it in the night when you want to sleep, you will all fit inside the bag, but in the morning again, you will fold it and keep it back. So even the endoplasmic reticulum is not very small, but it's because just it is folded. So because of this folding, it fits inside the cell. In some regions, the ER is studded. In some regions, the ER is studded. ER is studded. When we say it is studded, we mean that it has projections, and these projections are just nothing but ribosomes. So ribosomes are attached onto the surface of this apparatus or this ER. Where the ribosomes are attached, we call this rough ER. Rough ER or rough endoplasmic reticulum. And where we don't have them, we call it smooth ER. Smooth ER or smooth endoplasmic reticulum. So the surface of endoplasmic reticulum, as we have said, in the rough ribosomes, in the rough endoplasmic reticulum are attached to ribosomes. So as protein is being made on this ribosome, it enters the lumen. Inside the lumen, the protein may be modified by having sugar chains added to it, which can help the protein fold or give it stability. Not all ribosomes, however, as we have already said, uh, have these ribosomes attached on them. Some of them are just smooth. Some of them are smooth.